Hello everyone, myself HIMANI JAYESH from Shivajirao JONDHALE College of Pharmacy, Asangaon, studying in second year B Pharmacy. Today I am going to explain the theory of drug metabolism, that is first of B.Pharm, third semester. Topic name of theory is drug metabolism phase 1 and phase 2. Drug metabolism biotransformation. It is the type of chemical reaction which leads to modification of drug. Drugs are converted from one form to another to make them more active, less active, and finally inactive and to leave the body. Sites of metabolism: hepatic, microsome, mitochondria, cytoplasm; extrahepatic, lungs, blood, skin, GIT, and kidney. Microsomes, microsomal enzyme system, mixed function, oxidized monooxygenation. Its components include cytoplasm, P450 flow protein, NADPH molecule, oxygen. Mitochondria, monoamine oxide enzymes, cytoplasm, alcohol dehydrogenase, blood, stress amides, catechol, methyl transferases, install mucus and lumen, GIT flora, glucuronidase. Types of metabolic reaction. Phase 1 reaction: metabolic process that introduce or uncover polar functional group through oxidation, reduction, or a hydrolysis reaction. 2. Phase 1. Phase 2 reaction: metabolic process that involves conjugation of functional groups with endogenous polar molecule. Here we see the general pathway of drug metabolism. Characteristic of phase 1. First, inactivation, oxidation of paraorbital and alcohol hydrolysis of acetylcholine. Second one, conversion of active drug to another active one. Diazepam, oxydiazepam, codeine, heroin, morphine, phenylbutyrate, oxybutyrin. Third one, conversion of drug to toxic metabolites. Paracetamol, acetaminophen, halothane metabolite, hepatotoxicity. Fourth one, activation of prodrug. Chloride hydrate, trichloroethylene. Phase 1 reaction: make the drug more polar, more water soluble. Oxidation reaction: introduces functional group OH, NH2, SH, can be microsomal or non-microsomal. Microsomal: drug plus O2 plus NADPH plus H, change drug plus H2O plus NADPH. Example: first one, aliphatic hydroxylation, phenobarbital hydroxyphenobarbital. Second one, aromatic hydroxylation, phenoxetine. Third one, oxidation of amine anyl nitrobenzene. Reduction reaction: microsomal or non-microsomal. First one, microsomal nitrobenzene aniline. Non-microsomal, chlorohydrate trichloroethylene hydrolysis. Monomicrosomal only. Non-microsomal, oxidation by soluble enzyme in cytosol or mitochondria of cells. Example: 1-dehydrogenation and oxidasis. Second one, monoamide oxidase, hypotheine. Phase 2 metabolism. It involves union of the drug with one of several polar endogenous molecules that are product of intermediate metabolism to form water soluble conjugate, which is readily eliminated by the kidney or if the molecular weight is more than 300 in the bile. Phase 2 conjugation reaction. Example: morphine, paracetamol, and salicylate form conjugate with chlorogenic acid derived from glucose. Oral contributed steroid from sulfate. Phenylizine and dopacine are eliminated. Phase 2 reaction: almost invariable terminated biological activities. All is non-microsomal enzyme except glucuronidation. Characteristics of phase 2 products: morphologically inactive, more water soluble, to be excreted, more readily excreted in use. Modulation of liver microsomal enzymes: induction, inhibition. Liver microsomal inducer: alcohol, cigarette smoking, phenotype, sepromoin, grisophylline. Enzyme induction result in increased metabolism of the inducer, tolerance, decreased pharmacological action of the drug, increase the metabolism of combination drug, merbaritin, increase the metabolite mediated tissue toxicity, paracetamol and phenacetin as the therapy. Liver microsomal inhibitors, cimetidine, erythromycin. Enzyme inhibition may retard the metabolism and excretion of the inhibitor and co-administered drug. Prolong the action of the inhibitor and co-administered drug. Increase pharmacological activity. First pass metabolism. A drug can be metabolized before the drug reaches the systemic circulation, so the amount reaching systemic circulation is less than the amount absorbed, where liver, gut, wall, lung. Its result, low bioavailability, short duration of action. Factor affecting drug metabolism. Age. The enzyme system at birth, especially in preterm baby, are functionally immature, and especially for oxidation and for conjugation with glucuronic acid. The drug, like chlorophenolic, is unable to get conjugated, can cause fatal gray baby syndrome in neonates. After first week of life, the drug metabolic capacity increases rapidly. In elderly, metabolism is reduced because liver mass and liver blood flow are decreased. Metabolic inactivation of drug is low. Drug persist for longer time and in higher concentration the must be lower. Example: tricyclic antihypertensive, antihyperthymic drug. Enzyme induction process is also lessened. In pregnancy, hepatic metabolism is increased. This leads to decreased clearance of drugs such as phenothiazine and theophylline disease. Acute inflammatory disease of liver and cirrhosis affect function of hepatocytes and blood flow through the liver. This results in increased systemic availability of drugs such as propofol, labetalol, especially which has normally high first pass metabolism and exhibit prolonged half-life and reduced clearance. Drug metabolism is accelerated in hyperthyroidism. In food, some specific dietary factors induce drug metabolism enzyme. Example: alcohol, charcoal, griller beef, cabbage. Protein malnutrition reduces hepatic metabolizing capacity. I am thankful to our President, Honorable Dr. Shivajirao S. JONDHALE Sir, Secretary, Honorable Dr. Geeta Khare Ma'am, and Principal Dr. Nitin Mohere Sir, for encouragement and guidance of efficient use of social media to spread knowledge through the making informative videos. So please like and subscribe our channel for more informative videos. Thank you.